Welcome to the Tree Surveyor demonstration video. Okay, to begin we need to make an analysis project. We just click the new project icon here. There's a few icons here. New project, you can open a project if you've already saved something in the past, save project, or remove a currently active project. So we'll make a new project. I'll just give it a name. Whatever. Now we can see the new project has been produced. We can click that process and add a new process to the project. We have tree analysis processes for just general tree-like structures, whether they be hollow trees or hollow branch trees or solid. Kidneys and lungs. I'm going to load in a simple kidney E12.5 sample today for this demonstration, so I'll just click kidneys and OK. Now we have a kidney analysis project which has been generated. So the kidney analysis process, we can currently see that there's no source data selected. We can load in two types of data. One type of data, which is a grayscale 8-bit bitmap format file dataset, and we can load it from either OPT data, which has been reconstructed using Skyscan's and Recon, which is pretty standard for OPT. Alternatively, if you have a Z-stack from confocal or other imaging technology, you can load in a Z stack from that as again a consecutive numbered bitmap sequence. So we'll load in an OPT dataset. So because we've used the nrecon log file, it automatically grabs all the index positions and the image resolution and the fact that it's microns. But if it's an a confocal data set, then we can manually enter in an image resolution and units. Okay, so now that the data is loaded, we can see here in this kidney sample processing box is a grey outline around this box. So all this information in here is to do with setting up the processing. By default we have median filtering enabled, which is a 3x3x3 voxel cube filter. We can also, and that's by default, that's set, and that's pretty good for just standard OPT data, which sometimes has a little bit of noise come through from just a general byproduct of tomographic reconstruction. If your dataset is excessively noisy, you can click this, and that will produce a 5x5x5 filtered data set for analysis. But usually 3x3x3 is fine. This tool here, Enable Contrast Enhancement, is good if you've got a tree that may have been stained with an antibody and the intensity of the signal isn't uniform across the whole sample. Maybe the tips are weaker or some of the branching's weaker, depending on the expression of the marker you're using. Using this, we can do basically a local contrast enhancement, which will more or less normalize the contrast across the sample. That can be quite useful, but the caveat for that is that it can also bring out further noise, so by default we don't normally use that. So the cropping background threshold is used by default at 40 levels above black. Because it's an 8-bit bitmap, we have 0 to 255, and usually that's pretty good. But if you've got a particularly dark data set, you may need to reduce that a bit. So the minimum feature and maximum feature diameters are the diameters of the tree that you're looking at. So in this particular sample, so it's measured actually measured in voxels, but it's calculated as microns. So, well, whatever your data set unit is. So here I'm going to go 1, 2, 3, 4, 5, so 6 or 7 voxels, which is about 22 microns, and the maximum feature I'm just going to push out over here, which doesn't really matter too much at the moment. Okay, so once that's done, we can also set this skeleton detail extraction control, and that basically allows us to set the strength of the Gaussian filter that is used, which is really the core difference between this particular piece of software and any other that's out there that tries to do skeletonization. So we can use quite a broad filter by default, but we can use a finer filter too, a reduced filter, which is actually useful if you've got quite fine tips. And this data set that I've got does have some quite large and quite fine features, so I'm actually going to push this right out here to this -3 position. And once you've got that Gaussian filter data set, we don't normally see this, but internally in the software, once we've got that, we actually measure the strength of the Gaussian field, and we can either use many seeds to determine that strength, or many seeds to determine that strength, or fewer. By default, we use fewer. It reduces processing time, and also, we don't need to use many seeds to define a pretty decent data set. But in this particular one, I'm just going to increase it to 0.7. Okay, so with all that set, I'll just turn off this convex hull calculation. That actually defines surface area based on all the tips minus the root of the tree that's later set. The calculation takes a little while, so I'm just going to disable that for now. You can always go back later, remove that tick and continue, so we can do that later. Okay, so I've made some settings, so I'll just save that project again, and we'll execute the selected process to run it. So the processing now will go through median filtering. It does a basic grayscale watershed filling, then it performs all the Gaussian filtering. Once it's done that, now it's actually looking at all the seeding and locating convergent points. This all actually happens pretty fast on a data set of this size, which isn't overly large. Doing all the cost calculation functions. And once this is done, the following stage is the skeleton curation, which will happen just after this point allocation that's happening now. The software will actually use multi-core processors. Not all parts of the software are multi-threaded, but some that are used, so there are some benefits to using a modern AMD or Intel processor to access that. There is an Intel math library that's used for a lot of the 3D calculations, so there is some benefit to using an modern Intel processor. Okay, so once that's done, the first stage of the process is complete. There is no root set yet, so we need to do that. And also, if you want to make any modifications to the skeleton, we can do that in here. But first off, we'll just quickly look at the initial skeleton, which we can do by right-clicking the process, and we can view all the different 3D metrics that have or measurements or processes that have been done. So if we just look at the initial skeleton, we can see the volume in grey with the skeleton underneath in this yellow. In the instruction manual that I've written, you'll see that you can actually modify the view in this by pressing V on the keyboard. So if I press it once, it'll sort of give you the shade. Twice, it will give you an ISO volume of the data, everything above black, and then another view where it sort of reduces the intensity of the volume, so you can really see the tree. And that actually looks pretty good. Okay, we'll just close this, and we'll just jump over to the skeleton curation. So here in the skeleton curation box, I'll click modify. So here, the yellow skeleton, which was just rendered as a volume, is now rendered as cubes, 3D cubes. So in here we can actually set the root, which is the primary reason we're in here. So I'll just select the terminal root voxel, which is important. press spacebar, that becomes large, means that it's now set as the root. But any voxel you click will become white, and that's the active voxel. So if there are any edits that need to be made with the tree, this one looks pretty good, so I probably don't need to make any, or maybe not. So I can select a voxel, press delete, and that will delete a voxel. By doing that, I've now separated the tree here, so everything that's individual connected voxels are labelled in different colours. So here we've got a purple, there we've got a blue. If I delete another one up here somewhere, or down here, we'll see now that one's green. I can delete a connected group of voxels by selecting one of the group, doing Control and Delete, and they'll all go. I can also hold down Shift and click a voxel to select a second voxel. Then by pressing Insert, I can fill in between those voxels. That's the basic editing. We can also add in voxels. So for example, if we weren't satisfied with the way a tip was calculated, for example, say this one here that's right next to us now, I might select that tip, and then I hold down Control and click away from that tip somewhere, and we'll see where it's grown. Here we go. And if we're not happy with that branch position, for example, I could delete there, just delete that group of two with Control Delete, and I'll select that one and that one, and I'll join them, and now I've got a new position. To ensure, it's really important to ensure, if that is done, that you've not broken the boundaries of the volume. We can do that by pressing V to look at the binary and look at the position, the way the actual model underneath is, the volume underneath is curved, and that looks pretty good. So I'll just press V a couple of times to get out of that. Okay, so we've done some simple edits, and we've set a root. So we click OK. While I'm here quickly, a lot of the commands that I've just done are listed here in this information box down the bottom. So click OK. Now the edits that we've made appear in this modifications box. So we've deleted some things, connected and extended. Okay, so once we've done that, we'll just save the project so those modifications are saved, and we'll run it again just to commit those modifications. It doesn't go back right to the start and run it through everything, it just does the components that need to be done to finish it off. Okay, so once that's done, we have more or less a corrected skeleton which we can visualize in here, and really the only difference we probably would have seen from before is this little one here that we just tweaked. Okay, so we can also use a segment editor which we've built into the software. We have some corrected segment data now. We have some data now based on segments because we've modified the skeleton and committed the position of the root. I'm not going to view this. If you do view this, you'll see individual segments are listed in their, visualized in their respective colours. Sometimes I find that for me it makes it a little difficult to see things, so we can alternatively just say no here, and we'll see it in grayscale like we did in the initial skeleton editor. Okay, in here we can actually fold back individual segments. So if we want to exclude an entire segment, we can do that. So terminal voxels and branching voxels are displayed in a larger size, about the same size as what the voxel was when we set the root in the skeleton editor. We can, by clicking those voxels, we can actually set a continuity property. So we've got an orange segment here with the terminus of that segment, that branch point being orange. If I click on that with my left mouse button, it now actually sets a continuity, so that now this is actually classified as a single branch and it has an offshoot. If I click that again, then it'll flick up to the other branch, and if I click it a third time, it cancels it. So you can just cycle through those by clicking. That's actually very useful in a lung where you might have a bronchus with, say for example, you've got an incoming a bronchus which is actually quite linear, doesn't bifurcate in the same manner that this does. We can actually come through and select and turn individual branch points into continuous segments. I'll turn all that off, and yes, back to what I was talking about previously. We can remove remove segments here. So I just control click the terminus and it's removed. If we do that, we have to remember now that we've actually got a single segment which is broken into two, we need to continue that. So now we've actually got one branch point. So during the analysis, this would now actually be seen as a bifurcation. Got an incoming segment, one branch going this way, one going that way. Done. If we want to undo any of those edits that we've made, I can control click that voxel again and we go back to where we were, and I just click that and now we're back to square one. So we don't really need to do many of those edits, if any, in the kidney samples. Sometimes we have there might be a little nub that pops off one of the internal branches, which happens from time to time, and where that's useful is you actually see the segments are differently coloured and a little box here, in which case you can just come in and just chop that off in here, which is quite handy. But no, we haven't made any edits in here, so I'll just cancel this. Again, like the skeleton editor, there's an information box down the bottom here with all the information that I've just mentioned, with all the clicking and everything. Yeah, one other thing that I haven't mentioned is you can actually shift click one of the voxels, the branching voxels, and the software will actually automatically calculate whether or not it's a linear continuation, and it will set all the continuity properties automatically. And you can also attempt to do it for the entire sample by just pressing A on the keyboard. But in a kidney it doesn't really make any sense, because the continuity is based on the incoming and outgoing angles, and here the angles are far too great because it's a bifurcation. If it was quite linear and one linear branch with several offshoots, then that would be useful. So I'll cancel that. Okay, so we can actually look at now the segments, so we can see how the kidney now has been divided up into individual components. We can do that by looking at the colours, and those colours represent how the individual tips and segments have been divided and how their volumes are more or less set. They also give you an indication of how the diameters are calculated for the most part. Now, all the angle or most of the angle and length metrics and curvature are done using splines, and splines are calculated based on the skeleton. And here we can see the splines, they're smooth. The skeleton are used as control points basically to give you a smooth representation of the tree, which is much more probably organic and representative. Our software also actually extends those splines out to a tip to give you the precise location of the precise distance to the very edge of the tips, as we can see here. So these little green nodules are the branch points and the termini, and the cones represent the angles. So this incoming segment here has an extension, and that cone there is the extension, is the incident angle of the incoming here as it would pass through, and these are the angles of the splines that come out of it. So the initial angles of those splines are what are referred to in the software results as local angles, whereas the point-to-point direct, if you draw a straight line between the nodes, then that's a global angle. Okay, just close that. So the other useful tool we can use, the visualization tool we can use here, is to actually view the measured data with names. So each individual segment in the export is given an ID, and we can view the IDs next to their segments in this particular viewer. So we can see the root here that's labelled, and here we can see how we've got segment 2 and then 2.1, 2.2, 2.2.1, 2.2.2 and so on. So the names are inherited, which is useful because if, for example, segment 1 and its children have some particular importance, maybe that's an anterior branch and 2 and its children are a posterior branch or component of the tree. If I have my data I'll soon export, is in Excel, I can sort for everything that's 2, so I can just look at the metrics for one half of the tree, for example. Sort for 2 and its children or 1 and its children. Or, for example, the tips of 2 and its children. Well, the tips within the 2 group or the tips within the 1 group. So it's actually quite a useful tool to look for any features that you may think might be of importance, and then subsequently use that to analyse or hone down your analysis for later on. Finally, the last spline viewer, we can see so this is the angles that have been calculated. So red are more open angles, green are more narrow angles. Look, it's quite useful for very quickly having a look at features within the tree. Here we can actually see quite easily that the 2 terminal branches of the tree on either side, the anterior and the posterior half, are more open, and these lateral ones are actually more shallow, so that might be something also worth looking at in further analysis. Okay, once we've done that, we can export data. I mean, we don't need to go through all those visualization stages to do this, but I've left it till last. So we right click on the process, as I've just done, and down here there's a whole bunch of export tools. So export the global summary, segmentation summary, segmentation to Newick, which we can use in tree viewing software, GraphML, and GraphML tessellated. Okay, they've been exported. We can also export some of the volumes that have been calculated through the process. So there's a little expand here which I've just clicked next to the process, and now we can see these are actually all the components of the calculation that have gone through. They've all been done. That's why there's a little tick next to the little function icon. Then we've got a folder down here with data files. If I expand that folder, we can see this is all the data that's been generated, and actually sits in a subdirectory from your source data that you loaded in initially. These are serial files, so actually have no real use in any other software, but we can actually export a lot of these. So, for example, if I wanted to export the corrected skeleton, I could right click on the corrected skeleton volume.dat and export 8-bit grayscale to raw. So that'll export it as a raw data set. And we can do that to a few of these. Some of them we can't because they have no real use. They're just either vector files with mathematical results in them or so on. Okay. Just quickly looking at the data. 12.5, we have an analysis folder. I did a kidney analysis, so it's titled kidney analysis. If it was just a tree, it'd be tree analysis. If it was a lung, it'd be lung analysis. But in there, these are all the data files, and then there's some of the exported files. So, for example, summary text data, the GraphML, and the Newick, which is able to be used in tree viewing software. So I'll just open up the summary text here. This is just a Windows Notepad, so it's just a text view, it's not a spreadsheet software. But here we can actually see that we've got all the segment IDs, the segment names, and these are the names that we just looked at in the viewer previously. The generation of all the segments, and the inverse generation of the segments, so it sort of counts from the tips all the way back. So it's good because it's agnostic in relation to different branching patterns. So, for example, in the kidney we just looked at, there's some lateral branches where the tips are actually occurring after only 3 or so generations, but the terminal ones, the tips are actually it's from the root about 5 generations out. So, using this, we can actually look at all the tips or the branches or the segments just before the tips across the whole tree, which is quite handy. Metrics we have: length, diameter, volume, curvature of the splines, the angles between the incident and the segment, the bifurcation angle on a local level, a global bifurcation, and a dihedral local and dihedral global. The dihedral angles I haven't previously mentioned, but that is more or less the progressive angle between two generations and how they rotate between generations. And there are also a couple of tags placed on each segment, whether or not it's an offshoot, which is generated by the segment editor. None of these will be because we didn't use it, but also whether or not it's a terminus or a tip. So these ones are no, these ones are yes. So we can filter feed all the yeses here, and we'll get all the tips, and we'll get all these numbers for all the tips. Or we could just filter, say in Excel, for 2 and all 2's derivatives, so we just get all the numbers from all the 2. Now that's really the power of this. The GraphML file is just an XML file that contains all the data in the summary text, as well as the coordinate locations of all the nodes, which is something the summary text doesn't have. Okay, that's Tree Surveyor in a nutshell. I hope this has been of some use.